Morning students, our today's experiment is Enzyme Kinetics. The effect of enzyme concentration on the reaction catalysis. Enzymes are the reaction catalyst of biological systems. They have high degree of specificity for their substrate. Enzymes function in aqueous solutions under very optimum mild conditions of temperature and pH. The study of reactions rate and the influence of changes in environmental factors such as temperature and pH, etc. is known as kinetics. Enzyme kinetics is that branch of enzymology which deals with factors affecting rate of enzyme catalyzed reactions. The most important factors are enzyme concentration, substrate concentration, temperature, and pH. Today we are going to see the effect of enzyme concentration on the enzyme kinetics. Now how we perform the experiment. Step 1. Prepare 2 test tubes, one blank and the other test. In tube 1, add guaiacol, HRP enzyme and water. HRP is horseradish peroxidase. In tube 2, add guaiacol, H2O2, that is hydrogen peroxide and enzyme. Adjust the reading to 0 absorbance with tube 1 blank and start the reaction by adding 800 microliter HRP solution to tube 2 and read the absorbance of the mixture at 470 nanometers at every 30 seconds interval for 3 minutes. Note down the readings and observe the increase in enzyme reaction. Step 2. Effect of enzyme concentration. Again, these are 2 types of tests. Perform 2 tests, one with double the amount of enzyme and another with half the amount of enzyme. Prepare blanks separately for test A and test B since it contains double the amount and half the amount of enzyme respectively. Test A. Double the amount of enzyme. Add the reagents in tubes as mentioned earlier, taking into the consideration of increasing the enzyme concentration. Adjust the reading to 0 absorbance with tube 1 blank and start the reaction by adding 1600 microliter of HRP solution to tube 2 and read the absorbance of the mixture at 470 nanometer at every 30 seconds interval for 3 minutes. Test B. Half the amount of enzyme. Add the reagents in tubes as mentioned earlier, except decreasing the amount of enzyme concentration. Adjust the reading to 0 absorbance with tube 1 blank and start the reaction by adding 400 microliter of HRP solution to tube 2 and read the absorbance of the mixture at 470 nanometers at every 30-second interval for 3 minutes. The reaction kinetics under 3 different conditions will be compared on the basis of their readings and analyzed to see the effect of concentration of enzymes, which are shown by the graph.